Welcome students. This is chapter 3 in microbiology, the cell. So you can see how cells, which are the fundamental units of life, they come in different shapes and sizes, and it's so interesting to see the morphology of them. The main student learning outcomes in this chapter is to study what spontaneous generation is. You'll be able to explain the foundations of modern cell theory. You will also be able to explain the key points of germ theory, of endosymbiotic theory, the unique characteristics of prokaryotic cells, and also the unique traits of eukaryotic cells. So let's see what spontaneous generation is. So even like before Christ, before BC, like Aristotle, who lived in 300s BC, he is thought to be the father who started this thinking of spontaneous generation. It's this notion that life can arise from non-living matter. And then there's this famous experiment by Francesco Redi. So the experimental setup, it has these containers with meat in it. And if the container is open, the flies come sit on it. They lay eggs, and larvae and maggots will develop in it in an open container. But if it's sealed tightly, either with a cork sealed container or with a mesh, because the flies were not able to get in, there was no maggots in the meat. So he demonstrated that maggots were the offspring of flies and not spontaneous generation. So, and then John Needham comes and he does a few experiments, and he has this broth, but it's not sterilized completely. So what happened was microbes started to grow on it. So he offers an argument for spontaneous generation. And then Lazarno comes along and he disapproved Needham's findings. However, the most compelling evidence comes from Louis Pasteur. So he has the special flask. So this flask, he takes a broth, and this flask has a swan neck. So when the broth is cooled, it still remained free of contamination, because all the bacteria in the air and other microbes, they kind of settled in the curve of the flask, and it did not reach the broth. So however, when he broke off the swan neck of the flask, bacteria started to grow, because it allowed the air to enter the flask and contaminate the broth. So he definitely refuted the long disputed theory of spontaneous generation. That's why he's known to be the father of modern microbiology. So if you could recognize this word pasture, like from pasteurization of milk. And he also was a pioneer in vaccines and fermentation, like wine and beer, and how food spoil and all that. So we'll then see what cell theory is. So what is cell theory? Cell theory states that all organisms are made of cells. And cells can only come from pre-existing cells. So that is called biogenesis. It is the fundamental unit of all life, of all living organisms. So Robert Koch first observed cork cells under the microscope, and then he coined the word cell because it looks like a jail cell, like little cubes. And other famous names would be, so if you study the history of cell theory, Robert Hooke, coins the word cell because he saw cell-like structures in the microscope. Anton Leeuwenhoek also studies pond water and stuff, and he discovers bacteria. He identifies and describes bacteria and also simple microscopy. So it's only after the discovery of the microscope, everything like turns on. So Schleiden, he made observations to say all plants are made of cells. Schwann, who is a zoologist, he says all animals are made of cells. And Virchow proves that cells originate from pre-existing cells. The next thing we have to understand is the endosymbiotic theory. So what this theory says is, if you take a eukaryotic cell, the mitochondria and the chloroplast, which used to be a prokaryotic cell, they were somehow invaginated. They were brought inside the eukaryotic cell, and they established a symbiotic association with the eukaryotic host. So a pro-eukaryotic cell, so it didn't have mitochondria and chloroplast. So the cell membrane invaginated, and it captured a mitochondria prokaryote and a chloroplast prokaryote, a photosynthetic bacterium and an aerobic bacterium. So it is a symbiotic association. There are many scientists who worked on this, but Lynn Margolis, she worked in UMass in this proving this hypothesis. What are some of the evidence for this would be genetic sequencing and phylogenetic analysis of mitochondrial DNA and chloroplast DNA. So when they sequenced, even though it's an organelle in a cell, they have its own DNA, and it looks very much similar to a bacterial DNA. And they sequenced it, and they saw that it was very similar, both in DNA sequence and chromosome structure, like a plasmid, like a circular DNA. The mitochondrial and ribosomal chromosomes are also very similar. And another interesting thing is that they would divide by binary fission, which is also the way a bacteria would divide. Because eukaryotic cells usually divide by mitosis, right? But here, these organelles, they divide by binary fission. So they are very, very similar to a prokaryote, thereby giving a lot of evidence for endosymbiotic theory. So how did they discover germ theory, that germs cause disease? So there's, again, several very important scientists, and each one of them contributed a lot. So the germ theory states that microorganisms known as pathogens or germs will lead to disease. So when you say germ, it is not only a bacteria. It can also be a protist or a fungi, or even non-living entities like virus, prions, or viroids. So here's a brief history. So Semmelweis was the first scientist who proved that hand washing prevented the transfer of disease between patients to patients. So if the physician washed his hands between his surgeries or deliveries, there was less infection and less deaths. Then came John Snow. He conducted studies on the source of cholera outbreak in London, especially epidemiology. And then he pinpoints the outbreak to one water source in London. Louis Pasteur is studying spoilage of food by microbes and also infections. Joseph Lister, Listerine is named after him. He developed procedures for proper care of surgical wounds and sterilization of surgical equipment. So he used a phenolic compound to sterilize the equipment, and then he found that it caused less disease. But the most driving evidence come from Robert Koch's protocol to determine the cause of disease. It is called the postulates. So what are the postulates of Koch? So this is the pathogen must be found in every case of disease. It should be found only in the diseased person and not in the healthy individual. The suspected pathogen can be isolated and we should be able to grow it in pure culture. A healthy subject that is infected with this pathogen must develop the same signs and symptoms of this disease as seen in number 1 here. And then the pathogen must be re-isolated from the new host, and it must be identical to the pathogen from postulate 2. So here they are pinpointing. He pinpoints that a certain microbe causes a certain disease, like for anthrax and TB and cholera. So here's an interesting timeline of how, when the different things were discovered. So ancient Greeks proposed the miasma theory. So this theory is contaminated air, like vapors, like when something is rotting and it smells and there's these bad vapors, can cause disease. It's kind of like connecting a disease to unsanitary practices. And then here Cook comes, he's the father of cell. He names the cell, he coins the word cell. Leeuwenhoek, simple microscope, he's observing bacteria. Snow studies the epidemiology of cholera. And then like there's people approving and disapproving the spontaneous generation here. But here Semmelweis demonstrates the importance of hand washing, and Lister is using carbolic acid or phenolics to disinfect after surgery. And then it's Koch and his postulates that a specific microbe is responsible for a specific disease. So if you take a prokaryote, like bacteria is a prokaryote, how does it differ from a eukaryote? So if you take a typical prokaryote, it will have a cell membrane, a cell wall. Sometimes it might have a capsule to protect itself or like to stay dormant. The DNA is just like hanging there in the center of the cell. It's called nucleoid region. It doesn't have a nuclear membrane. See? And the ribosomes are also floating around in the cytoplasm. It might also have processes like a pilus or a flagellum, fimbriae, and so forth. So this is a typical bacterial cell. So the main difference between a prokaryotic cell and a eukaryotic cell is that prokaryotic cells are very, very small. See, it's only 1 to 2 microns, whereas a eukaryotic cell can be range between 10 to 100 microns. Prokaryotic cells will possess a cell wall that will prevent it from rupturing, whereas in eukaryotic, animal cells do not have a cell wall. However, we do see it in plants. But the main difference would be the membrane-bound organelles in the eukaryotic cell, which is lacking in the prokaryotic cell. So the genetic material is not enclosed in a nuclear membrane. And usually the genetic material is like a huge loop. It's like a big circle that is folded upon itself. Whereas in eukaryotes, the DNA is organized in chromosomes. So what are some of the common morphologies of some of the prokaryotic cell shapes? If it is round, it is called as a coccus. Cocci is plural. If it is rod-shaped, it's bacillus. If it is a curved rod, it's vibrio. If it is a short rod, it's a coccobacillus. If it's a spiral, it is spirillum. Plural is spirilla. And if it's a tightly, if it's like, if the screw is very, very tight, it is called as a spirochete. Spirochetes would be the plural. And here's some examples for each one of them. We have streptococcus that causes strep throat, bacillus cereus that is food contamination, vibrio cholera that causes cholera, Haemophilus influenzae which causes pneumonia, Aspergillus is Aquaspirillum serpens. So this is a spiral bacteria that can be found in fresh water. Borrelia causes Lyme's disease, which is a spirochete. So there's an example for each one of them here. What are some of the cell arrangements called? If it is single coccus, it's called as coccus. If it's two, it is called diplococcus. A four is tetracoccus. A chain is called streptococcus. A cluster is called staphylococcus. A single one is bacillus, and a chain of bacillus is called streptobacillus. Tonicity. So because most of the microbes, it's very important for microbes to adjust to the tonicity of the environment in order to survive, because most of the microbes might be subjected to a adverse environment. So the ability of the extracellular solution to make water move in and out of a cell is called tonicity. See, so for example, if the strength or the concentration of the solution outside the cell and inside the cell is the same, it is an isotonic situation or isotonic solution. So there's no net movement of water. So the amount of water entering the cell and leaving the cell are same. Whereas if you should drop a bacteria in a hypertonic solution, that means the solute concentration is high on the outside and the water concentration is high on the inside. So what happens is water will flow from high concentration to low concentration. So it's like osmosis the other way. So thereby it loses water. On the other hand, if you should place a bacterium in a hypotonic solution, where the water concentration is high outside, it can take too much water. So these are the different tonicities, and the same thing is explained with actual concentration of solution. So isotonic is what's going out and what's coming in are balanced. Hyper, it will shrivel. In hypo, it can cause the cell to expand and lyse. So here, let's look at some of the organelles in a prokaryotic cell. So if this looks like a bacterial cell, the DNA it's found in the nucleoid region, and it's usually associated with some special proteins here. And the bacterial DNA is like a long loop, a circular loop. However, there are extra chromosomal DNA, which is called as plasmids, which are circular, double-stranded. And one interesting factor is that it will carry genes that will confer traits like antibiotic resistance. So, and then throughout the cytoplasm, you can see these dots, which are ribosomes. So prokaryotes have 70S ribosomes, whereas eukaryotes have 80S ribosomes. So it's referring to the sedimentation gradient, the size of the ribosome. So there is also a lot of various inclusion bodies because these single-celled living organisms live in a very unstable environment. They will store up nutrients when it's available. So there is something called volutin or metachromatic granules, which is a polymer of phosphates. Polyhydroxybutyrate is a phospholipid monolayer, uh, which is like a source of biodegradable polymers for bioplastics. It might also have gas vacuoles, which is an accumulation of protein-lined vesicles of gas to change the buoyancy so that it can, be buoyant in a liquid environment. Magnetosomes are magnetic iron oxide or iron sulfide. So this will help in their movement. It will line up in a magnetic field. Carboxysome contains rubisco, which plays a huge role in carbon metabolism. Endospores are a very unique structure. So the bacterial cells are usually in their vegetative states. But sometimes what they'll do is, in order to overwinter a harsh environment, they will form endospores. So here it says the DNA will replicate, and then it will form something called as a phosphor. So, and then the phosphor forms an additional membrane, then a cortex will form around it, a protein core forms around it. So in other words, it forms a very tight protective, like a capsule-like structure called endospore. So now this will allow the bacteria to grow in unfavorable conditions also. So like when the conditions are unfavorable, it will become dormant as an endospore. And then it can come back to life when ideal conditions are restored. So the process by which a vegetative cell will transform into an endospore is called sporulation. So these are the steps of sporulation. There's asymmetric cell division. Then the phosphor will be surrounded by a cortex, a protein coat, and so forth, until it forms the endospore. So this is to stay dormant until the conditions become favorable. And Bacillus cereus, Bacillus anthracis, Bacillus thuringiensis, Clostridium can form endospores. These can be very hard to treat. So plasma membrane. So this is the fluid mosaic model of plasma membrane. It is a lipid bilayer. So it is made up of phospholipids. So there's 2 layers of them. And it is fluid. So it's in dynamic motion, and studded in the phospholipid would be all these series of proteins, these cholesterol molecules. Peripheral proteins are sitting on the outside. Integral or transmembrane proteins will go all through the cell membrane. So the main nature of phospholipid is it is selectively permeable. So this plasma membrane is selectively permeable, meaning it will allow certain substances to enter and it will stop some substances from not entering. So it's like a security guard. It will monitor what is entering the cell. So the phospholipids are in constant flux. That's why it is called fluid. And because there's a variety of proteins and carbohydrates that are connected to it, see like a glycoprotein or a glycolipid, it is called as a mosaic. So talking about the transport of molecules over the plasma membrane, it can be a simple diffusion. So simple diffusion is the movement of molecules from high concentration to low concentration until equilibrium is reached. So it's passive. If there is a protein that aids this movement of molecules, it is called as facilitated diffusion. If the movement of molecule is against the concentration gradient, then energy is needed, and that is called active transport. So these two are passive transport, and this is active transport. So this is the difference between the cell walls of a gram-positive bacteria and a gram-negative bacteria. So the gram-positive cell walls are comparatively simple, and there's a very thick layer of peptidoglycan with a tetonic acid extending into the plasma membrane. So here we see the cell wall is very important for the bacteria because it's highly protective in function. And it's protecting the bacteria from harsh conditions of the outside environment. It is made up of a chemical called peptidoglycan, which is a long chain of alternating molecules of N-acetylglucosamine and N-acetylmuramic acid, NAG and NAMS. And they are cross-linked with a lot of peptide bridges. So because peptidoglycan is very unique to bacteria, many antibiotic drugs are designed to interfere with this peptidoglycan synthesis, thereby weakening the bacterial cell wall and then making it more susceptible to osmotic pressure. That's how our drugs work. In gram-negative bacteria, the tetrapeptide chains will extend from NAM unit directly in a directly cross-linked way. Whereas in gram-positive, the tetrapeptide chain are linked by pentaglycine cross bridges. So these two are varying in their cell wall structure. So here's gram-positive bacteria, and you can see how the peptidoglycan layer is seen after the plasma membrane. So plasma membrane and a thick layer of peptidoglycan. And embedded in the peptidoglycan are the teichoic acid molecules. Whereas in gram-negative, you can see the phospholipid inner membrane, and then there's a periplasmic space, then a thin peptidoglycan cell wall, and it has another outer membrane. So this actually has three layers: an inner membrane, a thin layer of peptidoglycan, and an outer membrane made of lipopolysaccharides. So it's very complicated. That's why the staining protocol for gram-positive and negative is different. We use a differential strain for that. And this is a very interesting picture that shows how drugs, because we know the morphology of the bacteria, we can devise very effective drugs. For example, bacitracin will act on the cell wall. Rifamphin will act on the DNA. So we'll study this in detail in the future chapters. What is an acid-fast cell wall? So some gram-positive bacteria, especially of this genus, Mycobacterium, they have mycolic acid found on the exterior after the peptidoglycan cell wall. So the acid-fast staining protocol detects the presence of cell walls that are rich in mycolic acid. So here they will use a dye called Cobble fusion dye. And you'll be able to see the acid-fast bacteria. The bacteria is also might be protected with glycocalyces and yes layers. So this is like a capsule that is organized. So it's a firm layer outside the cell wall, and it is made up of polysaccharides, or it is usually a protein. Outside, it's a So sometimes it is a slime layer that is tightly organized or loosely attached to the cell, and it can be easily washed off. So again, the slime layer can be made of polysaccharides, glycoproteins, or glycolipids. The glycocalyces will allow cells to adhere to surfaces, and thereby they help to form biofilms. So when you say biofilm, it is a colony of microbes that are found on surfaces. They can hold on to water like a sponge. They protect cells from predation, and they hinder the action of antibiotics and disinfectants. Whereas the S layer is composed of proteins and glycoproteins. So apart from the cell wall and other capsules, you might also see these protective layers. So there are appendages that can be seen. So here you can see a pili. Sometimes it's a fimbriae. So when you say fimbriae, it refers to short bristle-like protein projections from the cell surface. So now fimbriae will help the bacterial cell to attach to surfaces. So for adherence, and also for maybe for colonization, for infectivity, for virulence, while adherence to surfaces is important, like biofilms. Pila is very, very interesting. It's a longer and less numerous appendage. This can also help with attaching the bacteria to surfaces, but it has this important role, like F. pilus or sex pilus, can be used to transfer DNA between bacterial cells. Flagella is a structure that is used to move in an aqueous environment. So it's a spiral filament composed of flagellin protein subunits that will extend outward from the cell, and it can also spin. It will have something called basal body, which is like a motor for the flagellum and is embedded in the plasma membrane. And the hook region will connect the basal body to the filament. So the flagella arrangements can be varied. So if a bacteria has a single flagellum located at one end, it is called as monotrichous flagellum. And if it's found on either end, it's called amphitrichous. So if it's found like a tuft in one end, is a lophotriphus. If flagella covers the entire surface all around, it is petrichous. How does the bacteria move? So the movement is either described as a run or a tumble. So it can tumble along. So movement is dependent on the flagellar configuration. So they will move in response to stimuli like light, then it's called phototaxis, or a magnetic field, or chemicals, like it's called chemotaxis. So the bacteria will achieve directional movement by changing the rotation of their flagella. So in a cell with peritrichous flagella, the flagella bundle, when they rotate in counterclockwise direction, it will result in a run. So, and if the flagella rotates in a clockwise direction, the flagella is no longer bundled. So that will result in a tumble. So coming to the review of eukaryotic cell structure and function. So you can see that the eukaryotic cell is very busy, is jam-packed with membrane-bound organelles. So here you can see the nucleus, the nucleoplasm, nucleolus, a double membrane protecting the nucleus. So the DNA is protected in the nucleus. And when it wants to express itself, it will make a copy of itself called messenger RNA. And the protein synthesis will happen in the ribosomes here in the cytoplasm. The central dogma of cell biology, how DNA becomes RNA, and RNA is the template for protein synthesis. This Golgi apparatus, endoplasmic reticulum, mitochondria, centrosomes, all these cytoskeleton, the microfilaments, microtubules. So it's a busy place. It might also have a flagellum. It might have cilia, peroxisomes. So there are a lot of eukaryotic microbes also. So some of them are algae, protozoa, fungi, and helminth, which are like microscopic worms. Here's some examples of eukaryotic microbes. Alga, trypanosoma, vorticella, paramecium, and plasmodium. This is plasmodium. Sorry, this is paramecium. So the main function of the nucleus is that it hosts the DNA and it controls all the cell activity. It's the command center of the cell. So DNA is responsible for inheritance and heredity. It is protected by a nuclear envelope, which will monitor substances that go in and out. The nuclear lamina is the filaments that are found inside. The nucleolus is the site of ribosomal RNA synthesis and ribosomes. And the cell is full of endomembrane system, like the smooth and the rough ER, endoplasmic reticulum. So rough ER does not have ribosomes on it. Rough ER has ribosomes on it, and smooth ER is devoid of ribosomes. So ribosomes are the protein factories in the cell. Golgi apparatus will play a huge role in protein processing, packaging, modification, and also like it will direct the protein to go to different parts of the cell or leave the cell. Lysosomes will have like little pockets of digestive enzymes. Ribosomes are usually ATS and they are involved in protein synthesis. Peroxisomes play a huge, they produce hydrogen peroxide, which is a reactive molecule. And then that will help to break down uric acid and other unwanted things in the cell. They also have catalase that can degrade the hydrogen peroxide. They will also play a role in lipid biosynthesis. So here by fluorescent microscopy, the cytoskeleton of the cell is lighting up. So there's 3 of them. There's microfilaments, which is made of actin. It gives stability to the cytoplasm. Intermediate filaments will maintain the position of the nucleus and also for anchoring cells. And microtubules, it is made up of tubulin protein dimers. And they are the girders within the cytoskeleton, and it will play a huge role in the movement. So these 3 are varying in size. You can see microfilaments are 25 nanometers, and the intermediate is about 8 to 10, and the smaller ones, so they are classified according to the size. Mitochondria is the seat of cellular respiration or the powerhouse of the cell. So this is where most of the energy is made in the cell. So sugars are broken down in the presence of oxygen and aerobic respiration to release carbon dioxide, water, and energy. So it happens in the mitochondria. Whereas so mitochondria, if you see, it will have outer smooth membrane and the inner membrane. So it's double membrane. The inner membrane is thrown into folds called cristae. It will have a mitochondrial matrix, and there's a mitochondrial membrane space here. And the different steps of the cellular respiration will happen in different parts of the cell. Chloroplast. So here's a chloroplast. This is also a double membrane. It will have like a stack of membranes called granum. The liquid part is called stroma, and they are capable of harvesting light energy to start the process of photosynthesis, which is carbon dioxide and water coming together to form sugars and oxygen. How membrane transport in eukaryotes. So again, it's a safe fluid mosaic model of membranes where the phospholipid bilayer is in a flux studded with proteins. So when a cell wants to bring in materials from the outside of the cell to inside of the cell, if it is a liquid, it's called pinocytosis. Solid is called phagocytosis, cellular eat it drinking. And if a receptor and a ligand has to attach in order to engulf a lot of material, is called as a receptor mediated endocytosis. The extracellular matrix is also very important in a eukaryotic cell because it will help to bind together with other cells, like for adhesion, proliferation, differentiation, and also for cell to cell communication. So the carbohydrates or proteins or lipids that are attached on the extracellular matrix has vital functions. Some eukaryotic cells also have flagella and cilia for cellular movement. The flagella has a 9+2 arrangement of microtubules and dynein motor proteins. Whereas the cilia, which is very unique to eukaryotes, has a 9+2 arrangement of proteins, and they can cover cell surfaces. And it might also have a basal body, like we saw earlier. And so overall, we studied the different theories like germ theory, disapproving the spontaneous generation. We studied cell theory, differences between prokaryotes and eukaryotes, and also the structure and function of the prokaryotic and eukaryotic cell. And I hope that was useful.